My name is Suyanee Thongchot from Department of Immunology and CAT, Sirat Hospital, Mahidol University. Today I'm going to present one of my projects in the title Personalized Neoantigen Immunotherapy Platform for Breast Cancer. Briefly about breast cancer. Breast cancer is the cancer developed from the breast tissue that has a high evidence in female worldwide. In 2040, it is estimated around 1 million women will die with this cancer. The treatment for breast cancer usually include the surgery with the chemotherapy or radiation. Unfortunately, patients with breast cancer have developed their treatment response failure and have higher rate of recurrence. And now there are several new and ongoing looking for the treatment for breast cancer. And the immunotherapy is the new type of therapy which may lead to the better outcome for breast cancer patients. Sierra Center of Research Excellence for Cancer Immunotherapy, or we call in short CAT, led by Professor Pethai Yentit Sumanat. We try to develop the knowledge and technology use the immune cell-based therapy, including the CAR T cell, dendritic cell-based adoptive T cell, and also the peptide-based adoptive T cell. And today my presentation is focused on the peptide-based adoptive T cell, and the tumor antigen that we use is the neoantigen, which highly expressed in only the cancer patient tissue. Briefly, we identify the immunogenic potential of the neoantigen peptide from the breast cancer, the tumor biopsy, and the blood of the patient by using the whole genome sequencing, whole transcriptome sequencing, and also the HLA class 1 and class 2 typing. Second, we generate the neoantigen-specific T cell from the autologous PBMC of each breast cancer patient. And third, important, we develop the autologous patient-derived organoid, or we call in short 3D PDO, and use as the target for test the autologous neoantigen-specific T cell killing assay. What are neoantigen? Neoantigen are the unique molecules or the unique protein that highly expressed in only cancer cell but not normal cell. Neoantigen are the cancer-specific antigen derived from the cancer-specific somatic mutation that which may be the non-synonymous mutation or the gene fusion that have been shown to be the crucial regulator of the clinical response to the immunotherapy. And now there are several about the neoantigen vaccine treatment that show in several in the cellular and animal state. And now they are shown in the clinical phase 1 and phase 2 study. So taken all together, neoantigen are the promising target to develop the anti-cancer treatment in terms of the cancer immunotherapy. And this slide shows like a schematic guideline for treatment with neoantigen peptide in breast cancer patients. We start with the screen the neoantigen peptide by using the whole genome sequencing and whole transcriptome sequencing from the core needle biopsy and the blood of breast cancer patient. And then the affinity binding of the neoantigen peptide of interest to the HLA class 1 and class 2 were identified. The first step will take the time around 8 to 9 weeks. And then at week 8, the 3D PDO or patient-derived organoid will develop in this from the surgical mass. And then the week after that, we generate the neoantigen-specific T cell from the PBMC isolate from the blood collection at the follow-up time of each patient. And then we test the neoantigen-specific T cell killing to the patient-derived organoid. And In this guideline, we believe this application could contribute to like a rapid screening, rapid validation of the neoantigen peptide for the personalized targeting immunotherapy in breast cancer patients. Okay, and this show like the pipeline that we screen the neoantigen peptide, that we follow the protocol from the handle in 2016. We focus on the 9 to 11 amino acid as a short neoantigen peptide. Let's stick to the HLA class 1, and more than 13 amino acid like long neoantigen peptide binding to the HLA class 2. And you can see here we use like 2 steps of the filter recommendation listed here, and then we got the candidate neoantigen peptide for short and long peptide for individual breast cancer patient. But finally, we select only 4 for short and 4 for long candidate neoantigen, considered with the best MT score, the gene expression level, and also the overlapping of mutation between short and long peptide. Briefly, we activate the PBMC of each patient with the candidate neoantigen peptide with the 3 cycles of restimulation. Totally take around 21st day, and the results show that each patient have the patient-specific neoantigen profile response to candidate neoantigen peptide that you can see at the first. They have the interferon gamma spot after we export to the candidate neoantigen, and also we success to generate the neoantigen specific T cell that you can see after 21st day. It can increase the production of interferon gamma in all patients. And also we confirm that our candidate neoantigen peptide can activate the ability of the anti-tumor activity of the T cell from the PBMC of each patient. That you can see is increase the secretion of the interferon gamma, granzyme A, granzyme B, perforin, and also the granulolysin. And also that you can see it can increase the population of the T cell with the interferon gamma production, and also increase the population of the memory T cell and also the effector T cell. Finally, we try to develop the 3D patient-derived organoid from the tumor mass of each patient. And this you can see the morphology of the patient-derived organoid, and also we confirm with the H&E staining with the original tumor mass of each patient. And also we confirm the living cell of our patient-derived organoid by stain with the cell tracker that confirm our patient-derived organoid is the living organoid. In the right-hand side, you can see after we export this patient-derived organoid to the neoantigen-specific T cell that stain in the red color, you can see the neoantigen-specific T cell can destroy our patient-derived organoid. This confirms the activity, like anti-tumor activity of our neoantigen-specific T cell that we produce. Okay, finally, I try to conclude this project like I integrate the multi-omic analysis, including the whole genome sequencing, whole transcriptome sequencing, and also the HLA class 1 and class 2 typing to filter out the candidate neoantigen peptide, also short and long peptide, and also show the strong immunogenic potential of each patient for the function validation. And also we show the pipeline that we follow, the PVAC sequencing pipeline that show that our neoantigen pipeline can activate the neoantigen specific T cell to show the anti-tumor immune response. And also, we success to generate the autologous 3D patient-derived organoid and use as the target to test in the laboratory. And I don't know why this slide show like this one. Okay, okay. And okay, just to conclude that this is just only the proof of concept to show how we can activate and generate the neoantigen specific T cell in our lab. So finally, I would like to thank the grant NRCT and also thank for the support from Professor Chanitra Thuvajit, Associate Professor Piti Thuvajit, and also Professor Pethai Yenjit Sumanat for the kind support for this project. And thank you for your attention and all of your suggestions and questions have really appreciate. Thank you.
Thank you for a very good, nice presentation. And I know this is not easy, right? To get a good result in vitro stimulation. Yeah. My question is, did you have a chance to look at wild type peptide as well?
Actually, we have the list of the wild type of each mutant peptide, but we doesn't use it because of the limitation of the budget that we cannot do like a if we have, yes, but in this project we just use only the unpulsed peptide to like the control condition.
Azai, thank you very much for your presentation. And well, let me think of the question. Okay. I'm wondering, you said that you already identified the four peptides candidates, right? And I'm wondering if you know where those peptides are from, like which gene mutation or like which proteins are they originated from?
Sorry, I'm quite not sure that I understand the question or not.
These peptides are neoantigen, right?
Yep.
So in my understanding, it must have arisen from mutated gene.
Yep.
Somewhere.
Yes.
And have you done the identification?
Yes, we have the list of the gene name.
Oh.
This is here, and also we can check the function and also the like protein name of this gene mutation.
Oh,
yes,
okay. And so those are from 4 patients or just one?
No, it's just
patient?
one. This is a representative case, just one patient. Yes.
I see. And then each patient have specific sets
yes.
of those 4.
Yep. Yep.
Okay. Thank you so much.
Okay, so we run out of the time. So we thank you very much for your excellent talk. So we will move to another presenter. Please welcome Dr. Raju Vivek. His topic is the tumor-targeted photothermal immunogenic nanovaccine induced efficient cancer immunotherapy.
Good evening to all of you. I think this is might be simply different from the topic whatever we discussed past 2 days. But my field of research basically in cancer. But we know we are recently past 2 years I'm trying to work on this cancer immunotherapy. But there are so many topics which are already you people are discussed. But you know, my mind is fully feeding the CAR T. It is okay. That is an an ultimate target. But also there is some of the trick that you have to target the tumor cells and you can induce the immune response. So for that, I'm trying to develop some of the nanovaccines. And these vaccines, mainly made up of some of the nanoparticles. So that nanoparticles it is acting as a carrier for some of the immunomodulatory agents. Okay, so once these nanoparticles delivering these immunomodulatory agents, that will induce the activation of some of the immune system. So I will go to some of the important things. So this slides is showing that the tumor microenvironment is the is an area inside which the tumors grow, and it becomes a focus of immunotherapy research. You can see that there are not only the tumor cells, it is also showing some of the antigen presenting cells, and more specifically inactivated T cells, okay, and also regulatory T cells, some other immune cells, but not like every cells that are active. Yeah, coming to, there are several therapies which is used for cancer based on this immunotherapy, like adaptive cellular therapies, oncolytic virus therapies, and cancer vaccines, and targeted antibodies. There are so many things, but my area of research, which is mainly focusing on immunomodulators. So in that immunomodulators, there are several things that are there, like immune checkpoint inhibitors and cytokines, and also the agonists or adjuvants. So you can see that the cancer patients, when they go for immunotherapy by using the nanoformulations, there are several, so far there are several, nano formulations have been developed, like they are used for just for carrier, okay, but by using some of the immunotherapeutic molecules that may be delivered through the different routes, like injection or spray gel, or sometimes it's a transdermal path, okay. Okay, so how this nanovaccine can work? Okay, so this is a very important paper they were published regarding the nanovaccine for cancer immunotherapy. And here you can see that there are some of the different nanocarriers that may be synthetic or semi-synthetic or biogenic. But you can also add some of the adjuvants or some of the tumor antigens. Okay, so here you can see how this nanovaccine can activate once it is delivered into lymphatic nodes. Then it is targeting the immature dendritic cells that become mature. Then it activates the T cells by activating the effector cells, and then it will killing the tumor cells. Okay, so if you look at this paper, the tool-like receptor is one of the important receptor which is expressing in antigen presenting cells. There are two types. One is surface targeting TLR, tool-like receptors, and another one is endosomal TLRs. Okay, so the one of the task is to activate the endosomal TLR, tool-like receptor is an effective task. So based on that, we have my group already we have published 2 papers regarding the development of new vaccines using 2 different TLR based nanovaccines. Okay, so one is TLR 7 and 8 agonist, that is nothing but resiquimod, and another one is TLR 7 agonist that is used to activate the toll-like receptors. Once these toll-like receptor is activated, then ultimately it is leads to activation of immature dendritic cells to mature dendritic cells. Then further it is activating the T cells. Okay. Yes. And if you look at this paper in Nature Nanotechnology, they have clearly shown that a general strategy towards the personalized nanovaccine-based fluoropolymers, okay, for post-surgical cancer immunotherapy. Here, they were used, one of the most important antigen, model antigen, like it is a OVA. So if you look at the population of the CD8 T cells, there is a dramatic increase in the OVA, which is incorporated with PEI, polyethylenamine. So this was an amazing paper, which is giving the overall background about the nanovaccine and how it is inducing the immunotherapy in cancer. And if you look at this is an same paper, the OVA antigen that was used as a model with different, some of the ICG FDA approved, fluorescent molecules that can used for an theranostic purpose. So they were also used, the combination of ICG and OVA, which will, again, they will inject into the tumor microenvironment through the intratumoral injection. Then it target the photothermal and immunotherapy. So here I will tell you what is the importance of photothermal immunotherapy. That was, you can see, if you look at this paper published by National Cancer Institute, the near infrared photoimmunotherapy of cancer. So this is a review paper, and what they were very clearly saying that when we are giving the near infrared laser to the tumor cells, it induces the immunogenic cell death activation. So once this immunogenic cell death is activated, then these dying tumor cells can able to release some of the immunogenic markers that are nothing but HMGB1, ATP, and calreticulin. These are the markers which is released from the dying tumor cells. So these markers also can activate the TLR, but it is not effective, okay, because it is on a surface TLR receptors, but these receptors also can able to induce the immature to mature dendritic cells, and they will activate the T cells, and further it will killing the all the tumor cells. Then if you look at this paper, they will use, I already told the R837 is an agonist, and it is specifically made for TLR7. So if you use this, they will use a PLGA polymer. It is an FDA approved polymer. In integration of these molecules inside, and they will induce the photoimmunotherapy. So that is what I'm also getting the point from this. So far I have done 2 TLR7 and 8 activation, but one more TLR receptors is pending, that is TLR9. So that is what I just trying to use this TLR9 as an TLR9 receptor to activate the immune system. So I have taken a photopolymer, this PNI. It is a conjugated polymer. Usually it used for photothermal therapy. So I have taken this polymer for inducing the photothermal therapy. So then we have loaded with some of the ICG and CPG. So CPG is specific molecule which is targeting the TLR9. So then we have confirmed with several characterization techniques, and we have confirmed. Okay. So this paper, why, if you look at this paper, why we can use CPG? So it is an immunotherapeutic use, and it is especially in cancer therapy. It induces the tumor cells cytotoxicity through the NK cell activation, CTL and APC activations. So then we are confirming some of the photothermal property of the material by using NIR light, and we have made that, and also we have confirmed the photostability of the material with various methods. And this is also, we have there are some of the photostability and the releasing property of the material from the from tumor microenvironment, and also in the normal pH 7.4. And if you look at this in the stability property, after the NIR light radiation, the material still it's showing the stable nature, but not ICG, because it is in molecules which is not stable long time. And it is an theranostic material. It has photoacoustic imaging properties, and which will be used for imaging purpose, not only in vitro and also in vivo. And these are some of the in vitro studies we have carried out, because photothermal property of this material, it can induce the tumor cell death through the photothermal activation. So we have confirmed with the staining techniques, and you see live cells are green and dead cells are in red in colors. So this is an in vitro part. Then coming to the in vivo part, we have developed a tumor mice like U14. And tumor mice we have developed. Then we have injected the these nanovaccine. Then we giving the NIR light 808 NMR nanometer. Then we are checking the tumor volume, and there is a drastic reduction of tumor volume after the post-treatment days. And also we are checking the various histopathological studies, but it is not at all affecting any of the organs, because we are giving the target specific activity. And you can see that after post photothermal action, we also checking these ICD markers whether it is activated or not. We have checked. We found that the expression of HMGB1 and also CRT is high when compared to the other 3 treatments. And also we have quantified the ATP level in extracellular place, that there is a significant increasing in the ATP when compared to, yes, okay. And also we check some of them. Okay. So this is some of the works. Okay, this is a conclusion by scheme. So ultimately, we need to activate the tumor immune microenvironment by using the CpG, TLR9, by using this CpG agonist. And then the future directions, because we are coming from a university setup, but we do not use any patient samples. But future directions maybe go for the patient samples, and that can be used for TLR activations. So this may be a point that you can also try with some of the TLR agonists that may be effectively used for activation of immune systems. Yeah, so I thank.
Well, thank you,ค่ะ. Well, due to time limitations, we have to move on to our next speakers. And if anybody have questions, you can reach him here in our conference. So may I invite our next speakers, Dr. Chutipa Chiawpanit from Chiang Mai University. Please welcome her.
Okay. Good afternoon, everyone. My name is Chutipa Chiawpanit. I'm a postdoc from Chiang Mai University. And this topic that I'm going to talk today is about the CAR-NK cell to target the c-MET expressed on a CCA cell line. And this project is my PhD thesis. So start with our target cancer, which is a CCA. CCA is a bile duct cancer that mostly found in Thailand, especially the northeast region of the Thailand. And the incident is related with the infection of the liver fluke. Nowadays, the only curative treatment is just a surgical resection. But mostly of the CCA patients were diagnosed as unresectable and metastatic disease. So now all of the therapy for the CCA patient only prolong their survival rate only 1 year. So now we mention with this problem, and we want to prolong their survival more than 1 year. So we interest in an immunotherapy platform to develop and treat the CCA patients. Here is the CAR. I'm not just develop the CAR T cell, and I alternatively develop the CAR NK cell because the NK cells show a promising result to be an off-the-shelf cancer therapy, immunotherapy for every patient. Here, not only NK not just activate to the CAR molecule, NK cell also detect with the various mechanisms like activating receptor to activate more strong reaction, and also avoid off-target reaction by detecting the inhibitory receptor. So I think NK cell is an interesting model to develop the immunotherapy just like CAR NK cells. When we talk about the metastatic disease behavior of the cancer, one of the interesting antigen is a c-MET. c-MET is a kind of a tyrosine kinase receptor similar with the EGFR or VEGFR. And c-MET also, after the activation of the c-MET, it can transmit the signal to activate the cell proliferation and cell migration. Here, there is some research, they confirm that after knockdown the c-MET gene, the number of migrated cell and invaded cell also decrease. And in the CCA patient, the higher expression of the c-MET also related with the poor survival rate. So we are going to go with the c-MET as a target protein to develop the CAR NK cells. Here is the concept of this study. We not just start to the CAR construct, we also start to screen the novel scFv to target the c-MET first. Before we begin all of the assay, we start to evaluate the level of the c-MET expression in the 3 CCA cell line, including KKU055, KKU100, and KKU213A. And all of the results indicated that KKU213A showed the highest level expression of the c-MET protein, while the lowest expression of the c-MET was found in KKU055. Then we next start to screen the novel scFv anti-c-MET by phage display and by panning technique. And I also screen the binding affinity with ELISA technique. And finally, we got 2 candidate scFv, namely as scFv11 and scFv72. We also perform a molecular docking to identify the location of the binding between each scFv to the c-MET protein. And we also have scFv-A, which is a published sequence, and we use this scFv as a positive control. So I start to construct the CAR construct, which is a second generation of the CAR molecule, and then transduce into the NK92 cell use a lentivirus vector. And here is the transduction efficiency. After co-culture for 24 hours, we found that our CAR NK cells show more specificity and more activity in the CCA cell that show the higher expression of the c-MET, while the activity not significantly different in the KKU055, which show the lowest expression of the c-MET protein. For all of this experiment, I would like to thank everyone who were mentioned in the slide for everything to support this project. And now I am working on a cell engineering for cancer therapy research group at Chiang Mai University. And we not just perform a CAR NK cell, we just try to develop another type of immunotherapy to treat the cancer patient, and we hope that it can be used in the real life in the future. And thank you for your kind attention.
Thank you, Ms. Dr. Chutipa นะคะ But due to time constraint, you cannot accept any questions, so you can accept it around in the floor นะคะ So may I invite our last presenter for the oral presentation session นะคะ Inthu-on Kulma ค่ะ please. Well, give her a warm welcome.
Good afternoon, all audience. I am Inthu-on Kulma, a PhD student from Thammasat University. And now I would like to talk about development of adoptive transfer therapy harnessing gamma delta T cell and NK cell against cholangiocarcinoma. And this study was done in the collaboration with the Professor Yoshimasa Tanaka from Nagasaki University. Alpha beta T cell and gamma delta T cell are 2 subsets of T lymphocytes. However, they are different in many ways, including mechanism of antigen recognition. Alpha beta T cell recognize the antigenic peptide in the MHC class 1 or 2 restriction manner. However, gamma delta T cell recognize the antigen in the MHC unrestriction manner. And additionally, gamma delta T cell exhibit at least 3 modes of cellular cytotoxicity against the tumor cell. The first mode is NK cell receptor mediated cytotoxicity, and the second mode is recognition of IPP in the tumor cell in the butyrophilin Ta1 or 2A1 by gamma delta T cell receptor, and the last mode is recognition of Fc portion antibody bound to the tumor antigen expressed on the tumor cell. You can see in this diagram in mevalonate pathway, nitrogen-containing bisphosphonate inhibit the activity of farnesyl diphosphate synthase, and resulting in the accumulation of IPP in the tumor cell to the level that can recognize by the gamma delta T cell receptor, and Then activated gamma delta T cell can kill the tumor cell by inducing apoptosis and secreting the cytokine. In several studies, hyaluronic acids passed with the interleukin-2 and or interleukin-15 were used as a product to simulate the expansion of gamma delta T cell before transferring it to the animal model in human. However, due to the negatively charged from the structure inhibit entry to the cell, and this limitation need to be overcome. PTA is a new effective compound that used to simulate the expansion of gamma delta T cell, and due to its high hydrophobicity, PTA easily enters to the cell, where intracellular acetylate can change it into the active form, or we call TA, and this TA can inhibit the activation of phacinyl diphosphate synthase, resulting in the accumulation of IPP in the tumor cell, and to the level that can activate the gamma delta T cell. Moreover, PTA produce the higher purity cell number and cytotoxicity against the tumor cell than the using of hyaluronic acids. It has been found that administration of allogeneic gamma delta T cell in advanced state CCA patient increase the peripheral immune cell as NK cell and alpha beta T cell, and decreasing the size of the lymph node without adverse effect. Therefore, our study, we want to compare the expansion and immunological property between gamma delta T cell and NK cell, and to develop adaptive transfer therapy, harnessing the innate immune cell for the treatment of CCA. First, we isolate the human PBMC from the peripheral blood, and we simulate the expansion of them by using PTA plus interleukin-2 for 11 days. And we found that the proportion of gamma delta T cell were increased up to 96% after cultivation for 11 days. Phenotypic analysis showed that expand gamma delta T cell express the high level of NK cell receptor as NKG2D, JAM1, and however, they express the CD16 to the different level depending on the individual. To analyze the cytotoxicity of gamma delta T cell against CCA cell by using non-radioactive cellular cytotoxicity assay, PTA CCA cell were incubated with the European Chimeric compound, and after washing, the PTA expand gamma delta T cell were incubated with the tumor cell for 1 hour. After releasing of labeling compound from the tumor dead cell, the fluorescence were measured, and we found that all CCA cell lines were killed by expand gamma delta T cell in an E/T ratio and PTA concentration dependent manner. And to determine the NK cell activation of gamma delta T cell against CCA cell by using luciferase-based assay. In this assay, we increase the incubation time between tumor cell and gamma delta T cell from 1 hour to 72 hours. And we found that a higher level of E/T ratio and a long duration time of incubation time are required for expand gamma delta T cell to kill the CCA cell. To determine the ADCC exhibits by gamma delta T cell against the CCA cell. All CCA cell lines express the EGFR at the different level, and we select the expand gamma delta T cell which have a high level of CD16 expression to incubate with the CCA cell line, and we found that expand gamma delta T cell exhibits a high level of cytotoxicity in some CCA cell line. You can see in this figure TFK1, SKCHA1, and MCCHA1. Moreover, expand gamma delta T cell secrete the interferon gamma in the presence of PTA at the concentration of 1 micromolar when incubated with CCA cell line. Next, we use the interleukin-2 plus interleukin-18 to simulate the expansion of NK cell for 10 days. And the results show that the proportion of NK cell were increased up to the 94% after cultivation for 10 days. And the last experiment is determination of ADCC exhibited by expressed NK cell against the CCA cell. And we found that in all CCA cell lines, they were killed by expressed NK cell in an E/T ratio and cetuximab concentration dependent manner. And it is very interesting that expressed NK cell require a lower level of E/T ratio to kill the CCA cell than the expressed gamma delta T cell. So we can conclude that we successful to express gamma delta T cell and NK cell and simulate innate immune cell exhibit potent immune effector function against the CCA cell line. However, the stimulate innate immune cell need to confirm the immune effector function against the CCA cell in the animal model. And additionally, pharmacodynamic, pharmacokinetic, and safety profile of PTA need to be clarified. Due to the low expansion rates of NK cell, a new strategy to simulate the NK cell need to be modified. And this is all of my presentation. Thank you for your attention.